All right, so I think most of us would agree that the development of antibiotics was one of the biggest medical breakthroughs, maybe even in history. Beginning with World War II, when penicillin was introduced and saved countless lives of soldiers on the battlefield, and then had widespread use globally. This was really hailed for good reason as a miracle drug. From everything from infections like sepsis that would have been a death sentence to maybe common STDs like gonorrhea, these could all be fixed with penicillin. But even in these halcyon days of this new kind of miracle cure, there was still something lingering, that we still had this specter sort of resistance already appearing. Before penicillin ever went to market, there were already Staph aureus species that were resistant to this. So how could that happen? Well, long before humans were even a twinkle in the eye of the universe, bacteria in the soils had been carrying on this biological arms race for millions of years, producing these antagonistic molecules that we know as antibiotics to kind of gain a foothold over other organisms in the soil, to outcompete them for resources. And these other bacteria then evolved resistance to that slowly, a little at a time. Maybe lots of them died off, a few survived and passed those genes on. And bacteria responded by creating new antibiotics, and other bacteria responded to that by creating new resistance. And so this was all fine and good until we appeared on the scene and started selecting for resistance by the overuse and abuse to some degree of antibiotics. But where we really made this jump was when these genes were sort of weaponized or mobilized from the bacterial chromosomes onto something called plasmids. So up here you'll see a plasmid. And a plasmid is an extra chromosomal piece of DNA that can move around. And some of them give resistance to one drug or maybe two drugs, or this one gives resistance to 5 different classes of antibiotics all at once. So rather than millennia of evolution, we suddenly jump from, a worm to a mammal in terms of the abilities and the huge jump that these plasmids can make in the evolution of organisms. All right, so then we have this pressure to keep these plasmids, and this is where we get something I like to call the superbug. Now, the name sounds really cute, but don't be fooled. These things are deadly. Superbugs are what we call bacteria that have become resistant to almost every single class, if not every class, of the main frontline antibiotics. And shown here is a representation of super gonorrhea that is making its way around the globe. And that's what they call it, so the CDC refers to it as super gonorrhea. And what was formerly this kind of really a minor embarrassment of an STD that you could, from the first slide you could see you could cure with a shot of penicillin in 4 hours, is now, these strains are treatable only with the last line, one class of last resort antibiotics. So this is scary, but it's a lot more scary if it's something that causes a much more severe disease than gonorrhea. So something like sepsis or bloodstream infections, essentially. If a patient comes into the hospital dying of sepsis, they don't have time to go through treatment after treatment and have antibiotics fail. If health practitioners don't know about emerging resistance or superbugs that are on the rise, they can't prepare for them and they can't respond appropriately to the needs of their patients. So hopefully here's where we come in. In some of my previous work, and this is going to sound really glamorous, working with sewage, comparing this to the human gut microbiome, we found that sewage does a really good job of reflecting what the human gut microbiome looks like. So the idea that I had was that we can use sewage as a way to do a cheap epidemiological study by taking a single sample of pooled human gut microbiome from an entire community at once. We can look at the antibiotic resistance that they're carrying, and then we can figure out for a given community what the risks are of potentially different antibiotics. And the important part here is that we want to look at commensal bacteria as well as pathogens, because pathogens are not our only concern here. Okay, so think back to that plasmid I showed you with those 5 different types of resistance that they could get. It's not just bad bacteria that carry these plasmids. Good bacteria can pick them up just as easily. We have a lot more good bacteria than we have bad. And those are not necessarily enemies. Sometimes they share these plasmids with one another. So the problem is that we can have our good bacteria carrying this resistance and potentially sharing it with pathogens. But our bodies and the clinics are not the only place that bacteria reside, of course. So anywhere we have overlaps between humans and our bacteria, the environment, soil, water, farm animals, urban infrastructure, these are all potential reservoirs of resistance. And so this is where my lab and I sample. We sample sand from the beaches. We sample impacted recreational waters. We sample sewage and septic tanks and things like that. And we use a bunch of traditional microbiological techniques. We grow bacteria on plates, just like you would expect, exposing them to different antibiotics, seeing which ones survive. We also take a DNA-based approach. So we look for the presence of these plasmids. We look for specific genes that can confer different types of antibiotic resistance, and then we look for the presence of those genes on the plasmids to show that they can be potentially spread around and shared. And so it should come as no surprise to people that we have found some of them. We have found a lot of resistance, which is not surprising, and this matches pretty well with what we're seeing in hospitals when we compare it to their antibiograms that they've developed. But we don't want to just reflect what we're seeing. We want to be predictive. We want to create an early warning system for emerging resistance that is not showing up yet in hospitals. So one of these things that we're concerned about is Colistin. Colistin was a drug that kind of fell out of favor in the 80s because it's very highly toxic to people. But it got picked up for use in animal agriculture, particularly as a growth enhancer for swine farms in China. So they feed it to the pigs, it makes them get fat faster. But in 2015, they found resistance, Colistin resistance, on a plasmid in these pigs. Then, a few months later, they find it in commercial meat. A few months later, they find it in a patient in New York. So this spreads globally and very quickly. So we have also found Colistin resistance. We have found it in sewage. We have found it in the environment. We have found it in commensal bacteria, our good bacteria, and we have found it in pathogens. So it's here. So I'm predicting that, and this is so gloom and doom, sorry, but that it's only a matter of time before we have an untreatable Colistin-resistant superbug case here in Northwest Indiana. But my hope is that we can continue to monitor this and that maybe we can find the next superbug before it finds us.